A topic lecture is Biochemistry of blood. Consider chemical composition of blood, metabolism of red blood cells, molecular mechanism of hemostasis, effect of medicine on the blood function. Blood is fluid connective tissue. Blood functions are respiratory, transport, nutrient supply, excretory, defensive, regulation. Blood consists of plasma, red blood cells, white blood cells, and platelets. Red blood cells are an important element of blood. It contains 60% water and 30% conjugated protein hemoglobin. Red blood cells contain no cell organelles as mitochondria, Golgi apparatus, endoplasmic reticulum, nucleus, and lysosomes. Red blood cells membrane is permeable to water, electrolytes, sodium and potassium, carbohydrates. Glucose is transported through red blood cell membrane by facilitated diffusion through glucose transporters. Insulin does not promote glucose transport to red blood cells. In the red blood cells, about 90% of glucose is metabolized through glycolysis, forming lactate, which is extracted into blood. Anaerobic glycolysis is the only pathway that supplies the red cells with ATP. Anaerobic glycolysis generates 2,3-bisphosphoglycerate, which regulates the release of oxygen from hemoglobin. Anaerobic glycolysis generates reduced NADH for conversion of methemoglobin to functional hemoglobin. In the red blood cells, the pentose phosphate shunt is the exclusive source of reduced NADPH. NADPH converts oxidized glutathione to reduced glutathione, which protect red blood cells against hemolysis. Glutathione is needed for the elimination of reactive oxygen substance, peroxidized hydrogen. Glucose 6-phosphate dehydrogenase deficiency causes drug-induced hemolytic anemia. The average lifetime of red blood cells is 120 days. As the red blood cells disintegrated, the hemoglobin is degraded into globin, protein fat, iron, and heme. Heme is converted to yellow pigment biliverdin by hemoxygenase. Biliverdin is converted to green pigment bilirubin by biliverdin reductase. Bilirubin bound to albumin and transport to liver. Plasma consists mostly of water that contains dissolved salt electrolytes and proteins. Paper electrophoresis of serum proteins uses an electrical field to separate the proteins into groups of similar size, shape, and charge. Blood plasma contains three major protein groups: albumin, globulins, alpha-1, alpha-2, beta, and gamma globulins, and fibrinogen. Functional plasma proteins are transport, reserve, maintenance of osmotic pressure, provide viscosity, defense, coagulation. Pharmaceutical companies prepare from human plasma products such as albumin, coagulation factors, and immunoglobulins is called plasma-derived medical product. Hemostasis is the process that stops blood loss from damaged vessels. Hemostasis is a complex physiological process involving cells, platelets, especially but also fibroblasts, insoluble proteins factor coagulation inhibitors, and insoluble proteins extracellular matrix proteins. Homeostasis has three major steps: vasoconstriction of damaged blood vessels, temporary blockage of break by platelet plaque, blood coagulation, or formation of fibrin clot. Later, the clot has been removed to allow wound healing. When endothelial injury occurs, the endothelial cell stops secretion of coagulation and aggregation inhibitors. At the site injury, endothelial cells release vasoconstrictor endothelin-1. There are three phases of platelet plaque formation. Adhesion is mediated by a variety of different platelet receptors, including receptor for binding to von Willebrand factors and receptor for binding to extracellular matrix proteins collagen. Activated platelets release many factors that promote hemostasis, including adenosine diphosphate is a potent activator of our platelets, serotonin and thromboxane, who want to assist homeostasis as vasoconstrictors. Von Willebrand factor enhance platelet adhesion and aggregation. Platelet-derived growth factors promote wound healing by stimulating growth and migration of fibroblasts to smooth muscle cells at the site of injury. Thrombin activates platelets via protection-activated receptor on the platelet surface. Platelet aggregation is the clumping of platelets together using fibrin as the connective agent. Platelet aggregation completes the formation of platelet plaque. The blood coagulation mechanism is formed then the plasma proteins fibrinogen is cleaved to fibrin, which afterwards become cross-linked into a stable mesh. Blood coagulation has three stages. Stage 1, activation of prothrombinase. Calcium and phospholipids are required for the prothrombinase complex to function. Second stage, conversion of prothrombin to thrombin. Third stage, conversion of fibrinogen to fibrin. Injured vascular endothelial cells release tissue plasminogen activator. It cleaves plasminogen into plasmin, which dissolves clot. Fibrinolysis is controlled by plasminogen activator inhibitors and plasmin inhibitors. Vitamin K is an essential factor for hepatic gamma-globulin carboxylase with 8 carboxyl group to glutamic acid reduce on factors coagulation 2, 7, 9, and 10. Vitamin K epoxide reductase reduce vitamin K back to its active form. This enzyme is pharmacologically important target for vitamin K antagonists such as warfarin. Anticoagulants inhibit special pathways of the coagulation cascade, which happens after the initial platelet aggregation and leads to formation of fibrin and stable aggregated platelet product. There are three major classes of anticoagulants: coumarins, vitamin K antagonist, heparin and derivative substance heparin, direct thrombin inhibitors. Antiplatelet drugs inhibit platelet aggregation. This group drugs include arachidonic acid pathway inhibitors, such as aspirin, irreversible inhibitor of cyclooxygenase enzyme through acetylation. Small dose aspirin inhibits thromboxane synthesis in platelets, but not proteocycline synthesis in endothelium. Phosphodiesterase inhibitors. ADP pathway inhibitors inhibit the binding of ADP to its platelet receptor by reversibly modifying the platelet ADP receptor. Glycoprotein 2B3A inhibitors inhibit the receptor for fibronectin, fibrinogen, and von Willebrand factor. There are three major classes of fibrinolytic drugs: tissue plasminogen activator, streptokinase, and urokinase. They act on fibrin binding circulating plasminogen. Streptokinase is obtained from beta hemolytic streptococcus group C culture. Tissue plasminogen activator obtained by recombinant DNA technique from human endothelial cells culture and hence are non-antigenic. Antifibrinolytic drugs aminocaproic acid and tranexamic acid. Conclusion our lecture. Textbook for prepare practical class. Thank you for your attention.